Okay.
Okay.
Yeah.
Can I start now?
Yeah.
See everything.
You can start.
Okay. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum warahmatullahi wabarakatuh. Ladies and gentlemen, good evening for all people in the night time countries. Good morning for the people in the morning time countries. And good afternoon for the people in the afternoon countries. For all in the present here, speakers and participants, I am Dr. Mohanna Shaybani from UTHM University, member of American Society of Microbiology. I will going the welcome speech for today's season for our conference under the title of Global Outreach Program in Microbiology Malaysia 2020. It's my pleasure to walk you through the program, introduce the speakers and our delegates that come from all over the country today. To share their knowledge and enhance our mental horizon in the microbiology. For today, we invite Dr. Adel, our young ambassador of ASM Malaysia 2020, to talk for 5 minutes about ASM and UTHM. Welcome, Dr. Adel.
Okay, thank you so much, Dr. Mohanna. Before I have to start, I would like to introduce our Professor Dr. Radin Maya Safira bint Radin Muhammad to give just opening speech. I'm going to introduce here, just brief biography. Prof. Dr. Radin Maya Safira bint Radin Muhammad. She is an associate prof at Faculty of Civil Engineering and Built Environment. She is the head of Microbiology Research Center, MBRC. This research center is in a collaboration with ASM to conduct all of this workshop. I think I have to mute all, right? Dr. Mohanna?
Mute all.
Right. Okay.
Yes, you can.
احنا ما نستعمل فارحين مو فارحين كلهم خالصين لا نقصد ولا واحد.
We make mute but still have some.
ليش يا جماعه تهموني انا دائما ابلغ، انا ابلغ فوق صوتكم حد ما تبلغ.
Dr. Adel should unmute, I think.
Make mute all.
Sorry, yeah. It's not me. Okay. So now?
Okay now.
Okay now. It's okay? Can you, Prof, please speak? I want to see if the voice is okay. Can you hear me or no?
Yeah.
I can
Prof.
hear
Maya?
you.
Yeah, okay. You are welcome.
Yeah. Is it my turn now?
Yeah. Now we can hear you. You can start now, yes. You are welcome.
Okay. Assalamualaikum and good day to everyone. Good morning, good night, good evening, good afternoon for everyone around the world. So this is my name is Radin Maya. Now I'm the head of the Micropollutant Research Center, University of Tun Hussein of Malaysia. I would like to say thank you to Dr. Adel, Young Ambassador of the American Society for Microbiology, Dr. Mohanna, Dr. Nayef, and all the organizing committee for inviting me here to give this brief speech on the our event today. So welcome to all of you, the speaker especially, Associate Professor Dr. Parveen, Fatimeh Ipani, and all of you to being able to join us today. This is a very great opportunity for us to work what we call that, to enhance our understanding and research collaboration in microbiology. So here in Micropollutant Research Center, we are working on various types of especially on the environmental science, engineering, wastewater, grey water, also the integrated water resources. So we are much welcome if you have any other research collaboration, publication and mobility. If you want to discuss, you can contact Dr. Adel and also myself. You may also contact through our Facebook, MPRC, so that we can have a future work together. So I hope that you can able to join all of our program so that we can get the benefit and also work together to know more about the microbiology. So congratulations to Dr. Adel and also the organizing committee. So this is a very good opportunity to all of us. Thank you so much.
Thank you so much, Prof, for coming for our activity today. Before we are going to start with the speakers, our respected speakers, we are going to just give me 3 minutes to give you a brief idea about the ASM and about the UTHM. I'm going to share with you some of slides. This is our slide. Okay. American Society of Microbiology and the UTHM. This program is sponsored by using American Society of Microbiology in a collaboration with UTHM, University Tan Hussein in Malaysia, and with MBRC, our Center for the Research at our faculty. Okay, American Society for Microbiology is a professional organization for scientists that they work on virus, bacteria, algae, fungus, protozoa, whatever under the term of microorganism. This organization has been founded by 1899. They have so many of free journals you can publish and you can also download and to find the recent attractive of the recent in American Society of Microbiology journals. ASM, they open membership for all the peoples around the world. For those coming from the development country in some of the country of the Africa, including also Yemen, the membership is free. For those they coming from developed country or some countries like Malaysia, they have to pay $100 per year. You can find more information about the membership by using the link. I'm going to provide for you both link for UTHM and for ASM in the chat. Okay, why we have to go in? Because you will get many of benefits from ASM. You can share with the information. You can publish in the journals is free. You can also find at the latest information about the microbiology in the journals. Okay, microbiologists usually they work in clinical microbiology, public health, biology, biological science, biotechnology, industrial microbiology, academia, different fields under the term of microbiology. UTHM is our university, is a public university established by 2007 as a full university. They have two campuses, Baguio campus and Barit Raja campus. UTHM is located as Guhur Baro at Guhur State, sorry, at Batu Bahat city. You can find more information. I will give you the link. Okay, UTHM they have several of faculty of civil engineering, faculty of electrical, faculty of computer, faculty of mechanical, faculty of science and technology and engineering technology, as well as diploma. This is the photo. You can see here the photo for UTHM. Okay, and this is the main campus. We have very big library with latest data with different database. So you are very welcome to our program today. I want to mention to you, we usually provide the link for registration for each sharing session. So don't just only look for the previous one. So you have to find the chat between time and time. We will give you the link. Please be confirmed that you join us and you record your name country, email, everything is right to receive our certificate. Thank you so much. And mic is going back again to Dr. Muhanna. You are most welcome, Dr. Muhanna. Welcome.
Thank you, Dr. Adel. Thank you so much for this short introduction about ASM and UTHM. For our activity of today, our speaker is Associate Prof. Dr. Parveen Fatima. Welcome, Dr. Parveen. Dr. Parveen?
Yes, Assalamualaikum.
Okay, welcome, Dr. Fatima. She will talk about hydrogen production and insights into thermophilic bacteria. Dr. Parveen Fatima is an associate professor at School of Engineering and Power Engineering, Jiangsu University, China. Dr. Fatima academic journey started from agricultural engineering in Abu Ali Sina University of in Iran. Later, she continued her master science studies in environmental science at Pune University, India. And therefore graduated from her PhD in Malaysia from Department of Environmental Technology, University Science Malaysia, Penang. Dr. Fatima continued her postdoc fellow researcher at Biofoil Institute, Jiangsu University, China, where she had professional work in biodegradation of lignocellulose waste through anaerobic fermentation using hyperthermophilic bacteria towards hydrogen production. Overall, Dr. Parveen is an environmental science with a rich academic background with over 10 years of experience in academia. Her main expertise is on waste management as Biofoil, focusing on lignocellulose degradation study. She has a good record for publication in well-known journal with over 700 citations. She contributed in a few books chapters and presented her work at international level. Welcome, Dr. Parveen. You can start and share your slides, please. Okay.
Is the screen is visible?
Yeah, yeah.
Okay.
Just yes.
Okay. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum, everyone. Very good day to everyone. Thank you for participating in this webinar today. Thank you, Dr. Muhanna, for this introduction. Also, thank you, Dr. Adel, for organizing such a wonderful webinar with us. And it is really great opportunity, and I'm really thankful to be chosen to be part of this talk. I'm sure this will be a good platform for us to connect, and it will be a good opportunity to share the information and bring us all together in the umbrella of science. So I would like to start my presentation today. I'm Dr. Parveen Rupani from Department of Energy and Power Engineering, Jiangsu University, China. These days, we are all talking about the renewable energy sources. Renewable energy sources such as biohydrogen, biofuels, bioethanol. These days, this topic has been received like much attention as a potential energy source to reduce greenhouse gas emissions. And among them, hydrogen is recognized as one of these potentials of environmental energy alternatives. Therefore, the topic for today, which I have chosen, is on biohydrogen production. This will be an insight into thermophilic bacteria. I will start the topic by having a brief introduction about bacteria, hyperthermophiles. We will have some, I will have some brief talk about biohydrogen production. We will lead the talk to an experimental study we have done here in China, and inshallah we'll close with the conclusion. So starting with bacteria. Actually, when we are talking about bacteria, we are talking about these small, tiny, living microorganisms which are abundant in our environment. There are many types of bacteria, of course, in the ecosystem, in our surrounding. Some of them are good bacteria and some of them are bad bacteria, but all of them, they have their own contribution in the ecosystem and environment. For example, there are some bacteria in different structures that we have, of course, like rod-shaped bacteria, such as Pseudomonas, cocoid-shaped bacteria, like Streptococcus, or spirilla shape. And each of them, they contribute with its specific role. For example, Pseudomonas can be used for wastewater engineering. Cocoid-shaped bacteria, Streptococcus, they are responsible for some of the human diseases. And spirilla also have been used environmentally in wastewater treatments. So actually bacteria, they have considerable diversity in shape, in their structure and metabolism. Bacteria cell wall, if we want to talk, it can be divided into two types, gram-positive bacteria and gram-negative bacteria. These bacteria, like gram-positive bacteria, the cell wall structure, of course, is very complicated. The main cell wall structure in bacteria is composed of peptoglycan. These peptoglycan are the main component of this cell wall structure of gram-positive bacteria. That's why if they want to divide it by staining, this will attach the color stain and turn to purple. However, in opposite side gram-negative bacteria, instead of having this full layer of peptoglycan, they have another outer layer as lipopolysaccharides. These lipopolysaccharides will avoid the staining process and it will remain as pink. Talking about this structure in bacteria, lipopolysaccharides in gram-negative bacteria actually is very important because this structure is in such a way that can help bacteria to resist in different environments, harsh environments, either in low temperature resistance, in low temperature, resistant in high temperature. And also they are resistant with many antibiotics. Therefore, studying about the lipopolysaccharides is very important. And it can be a good research platform for people that wants to study how this bacteria cell wall having this structure. For example, the lipid A structure of lipopolysaccharides, they are responsible for this resistance of into this environment. And if we want to talk about, of course, about the growth of bacteria, they will have different environments. They are growing, they have optimal growth in different temperatures. Like we have psychrophytes, which they will maintain the temperature, they grow in the -°C. Mesophiles, which mostly they are surviving in temperature of like human body. Thermophiles, the survival, the growth is up to from about 50 degrees. And focus on hyperthermophiles, if I would like to say, is the one that can grow about 80 degrees Celsius. There are many hyperthermophiles recently has been isolated, usually from hot spring, the hot plant industrial areas, to be used in industrial degradation processes. And specifically, I want to draw your attention to Thermotoga, one of these hyperthermophiles. These Thermotoga, they are the gram-negative rod-shaped bacteria, and they have many species under this Thermotoga. Like there's Thermotoga elfi, Thermotoga letinge, Thermotoga maritima, neapolitina, and so. And why this bacteria is important? Because they can resist, they can survive in high temperature, they are hyperthermophiles, they can resist in high temperature environment. So they have been preferred as a potential option for biofuel production in industrial application because they can produce high yield of hydrogen, of course. Continuing with this, among different species of Thermotoga maritima is recently a bacteria that researchers are working with it because of its many advantages. There are different researches have been done on different type of bacteria.
Dr. Adil, you mute all. Even Doctor, we didn't hear anything. Hello, Dr. Adil.
Do you have my voice? Sorry.
Yes, it's no, okay. Continue, Doctor.
So I was talking about T. maritima, that
Yes.
Recently the researchers have been focused on T. maritima, Thermotoga maritima, because of its various advantages. Like it has the ability of maximum utilization of cellulose and xylem. It has the ability of producing maximum hydrogen production rate, nearly to its theoretical value, which is 4 mole hydrogen per glucose. And also the stability of thermostable enzymes against denaturing conditions and extended reaction time. So these are advantages of using T. maritima over other Thermotoga species. Talking about the thermostable enzymes, actually, or I can say enzymes, is usually recently everyone there are many enzymes have been isolated from bacteria to be used in industrial applications. Like leather industry, they are using enzymes for degrade the byproducts from the industry. The dairy-based industries to degrade the whey products, already they are using enzymes. But most of these enzymes, more than 80% of them, they grow in a mesophilic temperature. While for the many of the industrial sites, there are problems that they need the enzymes which they can survive in harsh environments, such as high level of temperature, high level of pH, or salinity. Therefore, the importance of introducing thermophilic enzymes become very important to be used in industries. But the problem is the efficiency and the yield of thermophilic enzymes as it is very low. Therefore, the genetic expression method introduced into this area. And the best way is to clone this desirable enzyme into a common host, common mesophilic host to survive. And usually this mesophilic host been chosen by scholars is E. coli. Why they use E. coli? Because it has a fast growing characteristics, it's the cheapest way, it's a well-known host actually overall to maintain. Having further research, also they have seen that if using E. coli, there is a problem with it that during the process, maybe E. coli is producing a lot of lipopolysaccharides, which is producing the inclusion bodies in the system. And it is prohibiting for the efficiency of expression of this vector. Therefore, there are many host systems have been studied from different researchers, but it is very difficult for to choose a best hyperthermophilic vector for this purpose. However, in 2010, Professor Shao from School of Biofuels, Jiangsu University, she has worked tremendously on the thermophilic bacteria, and she could successfully make this vector gene expression actually by increasing the temperature, the growth temperature to 37 degrees. This way, she could use the heat shock method regulated by an alternative sigma factor of E. coli. And she designed this as a PHSH vector. So recently in our lab, we are using this method to make the plasmid, extract the enzymes, and use it in our system. This topic will be very broad, so I will just go forward to say more about the biohydrogen production. So of course, everyone knows that biohydrogen production will be having the process can be, the hydrogen can be produced by two main processes, light dependent and light independent processes. Light dependent processes is like photolysis, photofermentation, but the most focused and best process can be light independent processes such as dark fermentation. Because this dark fermentation is a process in which is economical, it can produce high yield, and through the hydrogenase enzymes, which is available in the process, in the system, the carbohydrate-rich products can be degraded and produce hydrogen for us. Many researchers have been conducted and proved that dark fermentation is the best process for using biohydrogen production, which during the pathway, the glucose through the pathway of pyruvates will convert to acetyl-CoA, and it can be, the output can be acetate or ethanol. In this process, the hydrogenase enzymes will help to produce hydrogen production. Now, if I want to draw your attention about T. maritima and fermentation. So as you see in this table, there are researchers that they have done research on studies on using T. maritima, but they use pure substrate for production of hydrogen. Of course, they are successful in it, but till now they are using the pure substrates such as glycerol, glucose, xylose, cellulose, and recently, maybe in 2018, they started to use on pure bio waste. They have used on fruit and vegetable waste, but still they have not challenged the lignocellulosic bio waste. Why is this important to use the lignocellulosic bio waste? Because the plant cell wall is mainly consists of cellulose, lignin, and hemicellulose. And this structure, the binding of this structure makes it very difficult to degrade, because lignin is resistant to biodegradation because of this cross-link network structure that it has. And penetrating through this lignin structure is very difficult. Of course, many pretreatment processes have been done, mechanical, chemical, biological pretreatments, but mostly these pretreatment processes cost maybe 40% of the total process. And beside that, if they want to use any alkaline or acidic pretreatments, again, they are posing addition of extra hazardous material waste in the environment. Therefore, always the challenge is to choose a green technology. For this reason, the bacteria comes into the picture. So if I want to combine all this information together, the experiment that we have done at our Biofuel Institute, we have chosen corn stover as a biomass material, lignocellulosic waste, and we have used Thermophilic maritima, which can grow up to 80 degrees. The special optimal basal medium also have been designed in our school by my professor, Professor Shah, and extracted this specific hyperthermophilic enzyme. We designed this bioreactor using our biomass, the bacteria introducing the enzyme. Of course, this will be an anaerobic process to degrade the biowaste and towards biohydrogen production. Through this process, we have obtained very good results actually. The hydrogen, we could obtain the hydrogen up to its optimal level. Near to the optimal level, of course, it wasn't full. The hydrogen was measured through the syringe method, and it was checked with gas chromatography. And as you can see in the picture, before addition of any bacteria or enzymes, we have put these reactors in incubator for 12 or 24 hours. And as you can see through the process from left to right, the color of the biomass is changing. It means it from this light brown, it is changing to dark brown. This means some process is happening, some degradation is happening, but there are some stickiness stuff is also visible, which we have to check for further studies. While we have extracted the enzyme from the cell wall of T. maritima, also we got a very good result, because we saw that the enzyme activities, xylanase especially, was very high in the cell wall process, and high protein concentration of xylanase and pectinase, which makes us believe that this definitely can be an approach to degrade the lignin structure of our corn stover. Also, when we have checked the enzyme activity, we have checked different enzyme activity, cellulase, xylanase, and pectinase, and we had industrial alpha xylose activity. But to our surprise, within 1 hour of experiment, everything was well, most of them showing increase, but after 1 hour, they are decreasing pattern. That's why this result and the results of previous slide, they are correlating with each other, that there is some inhibiting factor in our process, which makes us decrease in its activity. Either it is excess of hydrogen, which of course we try to maintain it, or lack of the food, or releasing some other substances which is unknown for us. Therefore, we continue the study to check what other monomers are releasing during the incubation time. And we have used HPLC method. Of course, because the monomers are low absorbance usually, and it cannot be detected in column C18. That's why we have used derivatization method. In this method, there is a label will be used, amino benzoic acid. This amino benzoic acid will make as a sheaf base, will attach to the to our sugar polymer, and makes the combination will be a secondary amine that can be detected in our HPLC result. And as you can see in this figure, the thick and the tall peak at time 35 is actually our label. This is the aminobenzoic acid label, ABA, which means that the process and the amination has been done successfully. Therefore, the peak beside to that, around time 30, it is our standard glucose standard. Therefore, we have run other standards, and we come up with this wonderful result. Apart from the glucose, which is of course in the process, it means that the structure is degrading, producing glucose. There are two other polysaccharides monomers we can see, we can detect, which is galactonic acid and galactose. Therefore, this can be an assumption that during this incubation process, apart from glucose, which is releasing, galactonic acid or galactose also is releasing in a good amount, which may making this inhibition factor for our process to decrease the process and decrease the hydrogen production. This results is till now, and it is still continuing. And we are trying to still battle this process and come up with the best result. So far, this is a very unique and very unique results we have obtained. So I welcome other researchers or scholars if they want to join our team, if they're interested to have further studies in this topic at Jiangsu University School of Biofuels. Very welcome. So to conclude and compile today's talk, we have told, or we have said that this degradation of corn stover using anaerobic hyperthermophilic bacteria, Thermophilic maritima, is possible without doing any pre-treatment process, we could see. And also the process can be enhanced while we have extracted its relevant enzymes. And more and most important one is to detect the inhibition factors such as galactonic acid. So it may be the reason for this stickiness stuff and decreasing our process. And of course, to apply its relevant enzyme, extract the relevant enzyme for further studies. As the future challenges, always we have future challenges in our environmental studies. So selecting and introducing this novel biomass degradation process via microbial consortia and enzymes, of course, to detect that which other pathways we can go. Like we can inspire from nature, having lignocellulose feeding animals we have in the nature. So we can detect the enzymes in the gut microbiota, detecting microbial consortia, effective enzymes, and towards, of course, having a green energy biofuel. So as environmentalists, we always think Thank you very much for your time and listening to my talk. And I would like just to close this talk with a short sentence saying that always be like eagle, be unique, and love to face challenges in your life. And most importantly, always. Thank you.
170. Now 30.
Thank you so much, Dr. Parveen Fatima. Thank you so much for your clear representation. Now we will open the session for answer questions. If there is any question, you can raise your hand, and Dr. Naif, you can select and unmute if you have any questions, please. What is our friend Dr. Ali Shahiri? He's chemistry. Dr. Ali? Ali Shahiri? Is there any question? Okay. Doctor, about the bacteria, do you determine the temperature? What we said, thermophilic means it is like bacteria temperature, high temperature. Do you know what's the degradation, the temperature which is degraded the cell wall?
Yeah, that's the research that we wanted to check. So now, of course, when you put it in a very high temperature, they will degrade. But this process is to put it in 80 degrees Celsius or 85, that they can maintain the temperature of thermophilic bacteria also.
So you optimize the temperature or not yet, the optimization of temperature?
Already it is optimized to growth for the bacteria. So the optimized temperature for this bacteria is 80.
Okay. What is about the difference between biofuels and the normal fuel from the petrol? Is this the same activity or how?
Different between biofuels and?
Yes, and normal fuels like, you know, the we get from the station. The
Of
quality.
course, biofuels, of course, yeah, of course, biofuels is based from biomass.
Okay.
So different generations by biofuels we have also, depending on the waste that you're using.
For the lignocellulose enzymes, we can get it from any other fungal, or just bacteria.
Yes, of course. From fungal also you can detect. There are many fungus that they are having this degradation ability. You can see it in jungle, woods. Specific fungi are working there to degrade the wood. So of course they can be isolated from wood also.
Thank you, Doctor. What is about our friend Prof. Abdurrahman Humaid? Dr. Abdurrahman, you're around?
Dr. Mohana, there's a question in the chat box.
Yeah, maybe.
One of the participants would like to know, apart from the corn stover, what other biomass that have you tried? Or do you suggest we can try, for example, bagasse, rice husk, or would conditions change? This is from Asha. Yeah?
Yes, of course, that is a good question. Of course, any lignocellulosic biomass can be checked. So far we have only checked on corn stover. And still, if this can be successful, of course, you can check with other lignocellulose biomass. As much as hard this lignin process is, you can penetrate the lignin, then you will say bravo. We can do for everyone, each ways.
There is one question. They said, how can we obtain the bioreactor? Use your experiment.
Say again, sorry. How can I?
Okay. How can we obtain the bioreactor used in your experiment?
The question is not clear to me, but the bioreactor which we use is like the anaerobic bioreactor, as I show in the picture, 100 ml working reactor. This is the lab scale experiment.
There is another question from Brother Nasser. He said, what is the yield that you may obtain from this process?
Yeah, the optimized yield is like, of course, we will wish to have at the optimal or near to the theoretical level, which may be very difficult. But as other researchers have been done up to 280 maybe mole per liter of glucose, perhaps we are also wishing to reach to that optimized good amount of hydrogen.
Okay. Thank you, Doctor. There is also a link for the certificate of today. Please don't forget to fill the form and you get your certificate for attendance for today. Thank you, Dr. Fatima, for your very nice presentation.
Thank
If there
you.
is any
Thank
question?
you, everyone.
Yeah, Dr. Mohana, there is some of a question also in the chat.
Okay, you can try and read because I don't see clear here.
Could you please, okay, there is one question here. Can we use such biotechniques in biotreatment of plastic residues?
That is really good question.
Yeah,
I wish.
he mean, can we use
Yes.
the plastic as biomass or substrate for biofuel?
Of course, we can use plastic, and recently, like the focus is, it is a really good question because now plastic is in the main focus also. But to degrade this plastic, there are researches have been done also, some bacteria they are using, but if the question is to then lead into the biofuel or biohydrogen production, it can be a good new approach.
Okay, there is one question. Thank you for your answer. For actually, so many questions here I can saw. Are you planning to try pilot scale for your experiment maybe?
Yes, inshallah, if we can make it successful to up to the the lab scale, Of course, this will be a very good opportunity to scale up as industrial scale.
Yeah, there is one asked about how we can isolate the erythema. Maybe this one is bacteria or what? I don't think so.
The same bacteria which is used.
Is okay. So isolation of bacteria is depend on the source. Okay, can we use such biotechniques? This one I already read it. Thank you. I have a question. Can we use such biotechniques in biotreatment on plastic? Already read. Okay, there one question from Esther here. If there is any side effect to human health after the use biohydrogen production, any health effect for the human?
It shouldn't be. That's why it is bio. That's why we are leading into the green technology.
Yeah.
So we want to replace it with the normal fuel because it's increasing GHG in the environment. So the main concept of this research is to reduce this effect.
Okay, Dr. Muhanna, come back to you. If you have someone want to ask.
There is no question. Thank you so much, Doctor. Thank you, all of you. Inshallah, for next season, it will be November 6th, Thursday, with Associated Prof. Dr. Iqbal Za'kan from Norway. It will be about natural antioxidant, natural defense against oxidative stress. Dr. Maya, there is any comments?
This is a very good sharing from Dr. Parveen. I'm very glad to have you here to join us today. I hope that we continue collaboration with Dr. Adel and Dr. Muhanna and everyone. Thank you so much. It's very nice.
Thank you, Professor. Inshallah.
Okay, thank you, everyone. Going back to Dr. Muhanna.
Thank you so much. Thank you all. Just don't forget about the link to fill. Can you get certificate? Saddam, Dr. Saddam Hussein, do you have any question?
Where is he?
No, thank you. I was actually in the training show. Just I come in the middle of this session, so I couldn't get the whole idea. Maybe next time, inshallah. Thank you so much, Dr. Muhanna.
Saddam Hussein, you can give us like, you know, he's a veterinary, surgery veterinary. You can give us like zoonotic microorganism, next time, inshallah.
Inshallah. Maybe, inshallah, if we have time, no problem. Thank you so much, Dr. Muhanna.
Prof. Abdurrahman is around. Abdurrahman Humaid. Dr. Khairullah Al-Ghazali.
Yeah, he's around.
Dr. Khairullah is our speaker. I don't know, it's not mute now. You can say no mute. You can open.
Dr. Muhanna.
Yeah. Assalamualaikum, Khairullah.
Alaikum salam.
So, do you have any comments, any question?
Thank you. Everything is clear. Thanks so much for the Dr. Fatima. Very nice presentation and very nice talk.
Okay, thank you, Doctor. Thank you.
Thank you, Doctor. Thank you. فيها.
Okay, thank you for all. We can now close.
Yeah, please, okay, Doctor, before closing, please don't forget to register using the link, please, all.
Thank you, Dr. Naif. Thank you so much.
Thank you. Well done.
Okay, so maybe
you will drop the link in the group of WhatsApp or what?
The chat.
No,
the chat.
The link here in the chatting.
In this chat, okay, just
chatting, please. Okay, I will share it.
Wait. Okay. You can share it in the WhatsApp. Okay, I find it. Thank you.
Yeah, okay. Thank you, everyone.
Thank you, Dr. Adel. Thank you so much.
Welcome. Okay.
Inshallah, our next season in the 6th of November with Dr. Iqbal.
Yeah, Dr. Parveen, she will be our moderator for that
yes,
sharing session.
Dr. Parveen, inshallah, our moderators.
Inshallah. Thank you. Thank you, Dr.
Well-
Mana. Thank you, Dr. Adel.
I will send you the CV and you can introduce here.
Okay,
Okay.
sure.
Thank you so much.
Okay, we have to stop now, right?
Yes, stop.
Okay, I will
No, no.
end all. Sorry.
Just wait some of. Okay, just share the link again.
Yeah,
Just 5 minutes.
Someone, yeah. Someone they will
Okay.
come to tell us, yeah.
Yeah.
So maybe we will give 5 minutes, but I'm going to stop the recording now.